Welcome back. In 1957, deep in the canopy of Karnataka, India, something began killing the wild monkey population. Soon after the monkey die-offs were reported, people living near the forest started falling ill with a severe hemorrhagic fever. Investigators isolated the pathogen and named it Kyasanur Forest Disease, though locals quickly came to call it monkey fever. The sudden canopy deaths were the first visible markers of a lethal virus spilling over from its natural reservoir directly into human populations. This map shows the specific altitude and forest areas of Karnataka, with the historical Kyasanur Forest Disease hotspots marked in red. For decades, the virus stayed contained here. Recently, it broke out, spreading across the Western Ghats into neighboring states. Currently, there is no specific antiviral cure for the infection. Medical care is strictly supportive. For years, the only line of defense was an older tissue culture vaccine. Due to genetic drift in the circulating virus and low overall efficacy, that vaccine was officially discontinued in 2022. Without a functional vaccine or targeted cure, the continued geographic expansion of the virus leaves millions completely exposed to an unchecked biological threat. At the microscopic level, Kyasanur Forest Disease is caused by a single-stranded RNA flavivirus. The pathogen relies entirely on an intermediate host. It does not spread from human to human. This diagram outlines the complete natural transmission cycle of the virus. The primary vector maintaining the pathogen in the environment is the Haemaphysalis spinigera tick. Small mammals act as amplifying hosts, breeding high viral loads. When ticks bite highly susceptible monkeys, it often results in fatal outbreaks. Humans enter this web strictly as accidental, dead-end hosts when they walk into tick-infested forests. Modern agricultural activities and rapid deforestation bring human settlements closer to these tick habitats, drastically increasing the frequency of tick bites. Clearing the forest strips away the buffer zone. It physically forces the human bloodstream into direct, daily contact with deep forest viral reservoirs. Once a person is bitten, the virus incubates for 3 to 8 days before launching its first attack. A sudden wave of severe chills, a piercing frontal headache, and intense myalgia, or muscle pain. This transitions into the hemorrhagic phase. The virus weakens the vascular walls, causing bleeding from the gums and nose, and severe neurological complications. For those who reach this critical stage, the case fatality rate hovers between 3 and 10%. The infection operates as a 2-stage biological assault. The initial phase disarms the host with severe flu-like incapacitation, allowing the second phase to systematically compromise the body's vascular and neurological systems. Following the retirement of the older formalin vaccine, scientists from Indian Immunologicals Limited and the Indian Council of Medical Research needed to build a modern replacement. This flowchart visualizes their solution, a next-generation whole virion inactivated vaccine candidate. The process uses an established varicella platform to produce a large-scale, highly purified viral antigen. This is then combined with an aluminum hydroxide adjuvant, a compound designed to strengthen the body's immune response. Modern vaccine engineering requires absolute, exact precision. Researchers must purify the antigen perfectly to trigger a massive, targeted immune response without accidentally causing the disease itself. Preclinical trials in mice showed that this exact formulation provided 100% protection against lethal challenges from the virus. The vaccine triggered strong IgG production, elevated T-cell cytokine responses, and generated neutralizing antibodies that successfully bound to the surface proteins of the virus, stopping it from infecting host cells. Based on the success of these animal models, the vaccine is officially cleared to begin Phase 1 human clinical trials in early 2026. A vaccine, however, is only one part of a broader strategy. This One Health approach integrates human medicine, active veterinary surveillance, and ecological preservation to track the virus across every environment it touches. While biotechnology allows us to develop effective vaccines, long-term survival against pathogens like Kyasanur Forest Disease requires us to monitor and respect the ecological systems we live in. Thanks for watching.